This is a production of Cornell University.
Well, it's wonderful to be here. It's wonderful to see old friends and colleagues that I'm working with now, collaborating, and also to be in a place with snow coming down. You know, I came from 9 inches and now I brought it with me, so you might get 9 inches. It's really a pleasure to be here, and I obviously didn't do the math very well when I accepted the International Plant Pathology Society presidency, because I'll be president in 2023. I'm president now, but it'll be 2023 when the meeting is, and obviously I'm not that young anymore, so I don't do math very well. But today I'm going to talk to you about something that I haven't often talked about, so a lot of people aren't aware that we're working in this area. But it stems from the fact that we're very interested in, I have been for my whole career interested in, so I started my career at Kansas State, and back then, for those of you who are young, it's a little bit different because when I went into Kansas, they said that I could work on any system I wanted to as a plant pathologist, understanding that as a plant-microbe interaction person, we were called disease physiologists at the time, that we would maybe choose model systems. And so I had worked in tobacco and Ralstonia solanacearum for my PhD, and then verticillium and hops for my postdoc. And I thought, well, I really want to work in, what a choice to have, to get to choose what you want to work with. I wanted to work on an important crop, and I wanted to work on important diseases of an important crop. So I cast around and asked questions, called my major professor, Luis Sequeira, and said, what would you do if you had it to do over again? He said, I'd work on Sigatoka in banana, or I'd work in Xanthomonas oryzae in rice. And about the time I started in Xanthomonas oryzae in rice, they had discovered race specificity. So the idea that there might be single dominant resistance genes. I knew that rice was a diploid, so it would be a tractable system relative to wheat, which is the state I was in. Kansas had a lot of wheat. And that the bacteria, Xanthomonas oryzae, that causes bacterial blight, would probably be genetically tractable. Back then it wasn't, but it probably would be at some point in evolution of my career. So I started working in this really important disease, which is still the major disease in rice, although Xanthomonas oryzae, as you'll see, is emerging as an important disease of rice. So the idea, my interest was, could I apply my microbiology and my plant physiology, back then we were called, skills from my PhD to solving this intractable problem? Well, we haven't done that yet, but you know, there are still jobs out there for people, because the issue really is that this disease is fairly well controlled by single gene resistance, and single dominant genes are sort of the mainstay for resistance. But the problem is, as you well know, in plant pathology, I don't need to tell people this, that you plant out sources of resistance, they're functional for many years, then you detect virulence in the pathogen population, the pathogen changes to overcome that resistance. So we have a continuous job. We have to keep identifying new resistance. In rice, it can take up to 10 to 15 years to find and integrate a new disease resistance gene. So if we're breaking down, as can occur within 5 or less years, it's not really a tractable system. We need to come up with other ways. So I've spent a large part of my career trying to figure out what I call the Holy Grail, and many of other people call it this too, which is how can we identify disease resistance that is not only long-lasting, but ideally effective against multiple pathogens, either multiple races of the same pathogen or multiple pathogens, like different bacterial diseases or bacteria and fungi or different fungal diseases. And I think the Holy Grail is a good example, because by definition, it not only provides happiness and eternal youth, which of course we'd all love, but it also provides food in infinite abundance. So today the story I'm going to tell you is a story about identifying resistance in different kinds of novel populations. And I'm going to tell you the story that Anna Bosa, her PhD work, where she was looking for resistance in rice that is effective against both two bacterial pathogens, which we'll talk about in a minute, so broad spectrum resistance, and specifically targeting Africa. And the reason is because both of these diseases are emerging as really important in Africa. And not only that, but there's no single gene resistance for bacterial leaf streak, so it's a problem. And they're being, because they're moving rice in from China into Africa, and those types of rice are susceptible to the African strains of these pathogens. And so they're having a big problem with the disease in Africa. And then I'm going to move into a second story because it uses the same magic population. I'll show you the power of this magic population by a postdoc in the lab, Alejandra Huerta, and Emily DeLorean, who was a master's student in the lab. So the two diseases that will be featured today are caused by Xanthomonas erysicola, which is bacterial leaf streak of rice. This is really an interesting disease because, and the difference between these diseases is really interesting. This is an intercellular pathogen. It enters the plant through the stomates and it moves in the plant in the intercellular spaces. Whereas Xanthomonas oryzae, very closely related genetically, is a vascular pathogen. It enters the plants either through wounds or the hydathode water pores, which are located on the edges of the leaf, and it causes a blight-like symptom. So two very similar pathogens, but two very different lifestyles. So finding resistance that's effective against both could be a little bit of a challenge, one might think. As I mentioned earlier, bacterial leaf streak is emerging as a major problem in parts of Asia and in Africa. It's actually thought that it used to be observed only on one side of the Yangtze River in China. Now it's observed on both sides of the Yangtze River, and they're thinking a lot of it is because of climate variation and impacts of weather on the system. In this case, we don't have single dominant resistance gene. The only single dominant resistance genes that we have to this pathogen are one that we identified from maize years ago, which is when introduced into rice, controls the disease. But otherwise, they usually use quantitative trait type loci or quantitative trait governed resistance to control this disease. So there are limited resistance sources for it, not only in Asia, but also in Africa. And in bacterial blight, as I mentioned, is a vascular pathogen. It's controlled by single genes. But there are few sources of resistance that have been identified that are effective against the African isolates of this pathogen. Oops. So let me introduce you to the population that we used to identify sources of resistance. So my colleagues at the International Rice Research Institute a few years back developed what are called rice magic populations. And these populations are multi-parent advanced generation populations. And folks may have worked with them, and there are now magic populations not only in rice, but also in wheat and other, also Arabidopsis. This population has 8 parents. So you can imagine the genetics that you're scrambling when you're intercrossing 8 parents, and we'll talk about how it's done. But I want you to notice this is the population. There are 2,000 lines planted out at Erie where they're propagating it. I want you to notice how even, how beautifully even it is. It's because in this particular case, 8 of the parents that were selected are elite varieties, but they have different traits of interest. So what that means, and importantly what that means, is that anything that we identify in this population is very close. It's like a pre-breeding material. It's very close to what the breeders can use to integress into other elite varieties. You don't have a lot of genetic drag from other things. So the other reason it's nice is because not only did they select those 8 parents for useful traits like disease resistance, drought tolerance, salt tolerance, phosphorus utilization, but the population is huge. There are 21,000 magic lines. So when you map in that population, you're almost getting down to the genes of interest. And the offspring, as I mentioned, can be integrated directly into breeding programs. So I'm not going to spend a lot of time on this because I'm not the person that developed it, and it's a pretty complicated process and long-term process. But you take the 8 parents and you do a series of crosses. And in fact, you've crossed at least 3 times to develop this particular population. And then after you've finished crossing, you start selfing. And then you self, in our case, we use the S8. So you get rid of any other extraneous information, but you have really a single, that is a true line once you get it. So essentially you have 21,000 isogenic lines once you get down to this. And they breed true because rice is a selfing population. So this is really powerful, but really they hadn't explored the utility of it for disease resistance. They had a little bit of work on that, but not much. So when Anna came into my lab, we wanted to ask the question of, could we use this magic lines, this series of magic lines, to identify sources of disease resistance that are novel and that are effective against both Xanthomonas oryzae, the bacterial blight pathogen, and Xanthomonas oryzicola, the bacterial leaf streak pathogen. And we focused on the African strains because we had this issue of the disease being prevalent in Africa, but no sources of resistance. So the first step was to genotype the materials, and that was done by genotyping by sequencing. So we have 14,000 SNPs that we were using, so not the entire suite of SNPs, but 14,000. And we screened a subset of the S8 population, so really fixed lines, and we did about 330 lines. And the importance of that is that's not a huge population to screen, but you'll see it's very effective. So we could identify sources of resistance in a very small population. We phenotyped with the 2 diseases. The vascular disease, we use a scissor clip to introduce it into the vessels. The intercellular pathogen, we infiltrate into the space. And so the experiment was done with 20 different, very diverse Xanthomonas oryzicola and oryzae strains, 10 each. And we not only did this in the greenhouse, replicated in the greenhouse over time, but we also, with our colleagues in Africa, screened in the field. Because obviously, if you're looking for resistance, it has to be effective in the field. Then all of that data is taken and integrated and analyzed. And because we're using a huge population with multiple parents, you can do GWAS analysis or genome-wide association analysis, as well as interval mapping. So what did we find? And I'm not going to give you the whole experimental process that Anna used to get to the steps because I want to move into the second set of work. But this is an example of one set of strains. You can see that the indica was segregating for resistance, and you can see typically that you have transgressive segregation where you have lines that are more susceptible than the 8 parents, and in often cases we have lines that are more susceptible or more resistant than the most resistant parent. And this is just 2 of the different pathogens that were used in the inoculation, one oryzae and one oryzicola. So that's good. We know that the population, oh, I forgot to tell you a very important point. the other good thing about the magic, because you're scrambling the genomes, you can do two things. You can create new novel resistances because you get recombination, and you can uncover resistances that you might not have seen in a particular parent because of its genetic background. So with a population like this, you have many obvious opportunities for creating new sources of resistance. So Anna, in the GWAS studies, was able to identify sources of resistance to not only Xanthomonas oryzae, bacterial blight, as you can see from this, but also when she did identify those sources of resistance, what was wonderful was, this doesn't always happen, and it's in general true, but not always true. We can confirm what we identified in GWAS by the interval mapping. That doesn't always happen, but that's a good indicator that what we're seeing is really real. And in fact, the most important piece of information is that we were able to recapitulate what we saw in the greenhouse in the field in Mali. So in the end, what she ended up with was a map, and that map includes resistances. So she identified 14 different QTL that were effective to multiple Xanthomonas oryzae strains. And both Xanthomonas oryzae and oryzicola, she got 11 broad spectrum resistances, that those regions conferred resistance to both pathogens with very different lifestyles. And then she was able to identify 3 pathovar specific QTL. So this is great, but what's most interesting is this one here on chromosome 4, because the stars indicate and the bars indicate different isolates. And you can see that the one on chromosome 4 hit a lot of isolates. So that is a source of resistance that we had never observed in any other population. And what's interesting about it is that when we look at that region, we can see, because we can map this with both GWAS and interval mapping, we can actually get down to what SNPs are important for resistance and which SNPs are important for susceptibility relative to the different isolates. So that helps us narrow into the region with the genes. And it was effective against many of the isolates that we had screened it with from the field, in the field and in the greenhouse. So a major contribution in that particular aspect. So to summarize this part, using the MAGIC population, which we didn't know if we were going to be able to see sources of resistance to either of the pathogens, really. I mean, there were a few single dominant resistance genes in the population. We saw them. But we were able to pull out 11 different genes that were effective against both pathogens, which is huge. And that one on chromosome 4 is one that we're focused on still. And again, it was confirmed in both the greenhouse and the field. Again, this is important because now that we have these lines with, for example, this chromosome 4 QTL or this region of resistance, we can begin to integrate this into varieties that are useful in both Africa and in Asia, for that matter. So with that as a backdrop, we now knew that this population was manageable, and we didn't have to screen all 2,100 lines in order to identify something. We thought, okay, let's start to see if we can begin to ask questions with that population that we've been interested in for some time. And so Alejandra led this project with the help of Anna. I want to point out mapping with 8 parents that are scrambled up is not trivial. And the programs, the analytical programs to do the GWAS and the interval mapping were just being developed as Anna was doing this project. So she was on the phone with this molecular mapper, the bioinformatics person who originally was in the Philippines, then was in Australia, and now is at a company in California. And Anna was continually, and they were wonderful at working with her. So she became a really valuable resource for the rest of the work in the lab and learned a lot more than she expected to about these populations. So the next question was, can we use this population to identify rice varieties that target specific effectors in pathogens? And why would we want to do that? Why is that of interest? So many of you, especially in this department where Adam Bogdanov is, know that tall effectors are the dominant effector families in xanthomonids. And in fact, Xanthomonas oryzae and Xanthomonas oryzae and Xanthomonas oryzae rhizicola have multiple copies of tall effectors. So these are transcription activator-like effectors. They're the major virulence factors for these pathogens. They have other effectors, but these are the major ones. The effectors are produced in the bacteria, pumped into the plant cell using a type 3 secretion system. Once in the plant cell, they're mobilized into the nucleus. They bind to the promoters of genes in the plant, and they activate the transcription of those genes. We'll come back to that in a little bit. The Xanthomonas oryzae strains that we work with have multiple copies of the tall effectors. They can have up to 20 or more copies of these effector genes. And again, they bind to these sequences in the plant and activate their expression. Now, there are several important factors here. Not all of the tall effectors function, and not all of them function equally in the plant. So a number of years ago, Jian Fabai, when he was in the lab, showed that if you knock out one gene, AVRXA7, you reduce virulence of the pathogen significantly. And interestingly, if you knock that gene out, it's a major effect tall effector. So in order to see the effect of other tall effectors, we had to work with that mutant and knock out other genes. So this is a double mutant. And you see that there's no additional effect when he knocked out AVRXA10. We continued to work with that particular set of isolates, and we also did field studies, Nali Vera Cruz, when she was in the lab, and we showed that at the field level, a number of years back in 2000, that if you lose AVRXA7 function in the field, not only do you lose the effector function, the avirulence function, but you also lose the fitness of the pathogen, or the aggressiveness and reduced fitness in the field. So it doesn't persist in the population. So it seems to be a very, very, very important fitness factor. So with that work, we had at that time proposed a hypothesis that you could predict the durability of a resistance gene based on its contribution to pathogen fitness. So AVRXA7 is a very important effector, and therefore if a resistance gene targets that resistance gene is probably going to be more effective and durable in the field than one that targets AVRXA10. And in fact, Nali showed that in field experiments and in greenhouse experiments, that if you lose AVRXA7 function in the field, if there's mutation in the field to that, you lose, the pathogen doesn't persist, and that is a very effective resistance gene. So we thought, well, there are other effectors in Xanthomonas oryzae, and we had looked at a number of these other effectors doing similar studies like this, and one of them stood out because it was called TAL7B. It's also a tall effector. It's found in the genomes of many different Xanthomonas oryzae. But this one has only an incremental or a minor contribution to virulence. So if you knock it out as well as AVRXA7, you get an incremental decrease in the virulence. So our question was, also this one is found in almost all the sequenced Xanthomonas oryzae genomes. So it's present everywhere. It has a minor contribution to resistance. So if we identify sources of resistance to TAL7B, could that be added to our gene pool for resistance to help stabilize and make the resistance more durable, since so many bacteria have it? Even if it's only contributing a little bit to virulence, is it contributing more to other things like fitness in the field? So Alejandra and her colleagues set up an experiment similar to what Anna had done. We took the genotyped lines, we phenotyped them. In this case, we subcloned and confirmed first that we had Tal7b. They put it into a strain where we were overexpressing it purposefully because it's a minor effect and we're screening a population. You need to have a pretty hefty gene product in order to do that. And then went through the mapping process. So importantly, the magic lines segregate for resistance. Initially, you can see that they segregate. We have a transgressive segregation again. We're seeing higher levels of susceptibility. So I'm going to talk to you in a minute about the function of Tal effectors, and so that's not surprising. But you have the population has lines that are more susceptible than the parent. That also implies, importantly, that you can map for susceptibility if you want to understand virulence in a population. The other interesting point to this is that we were able to identify resistance to both PXO99 with the empty vector. So this is the control strain. So we have lines that are more resistant than the parent, as well as lines that are more resistant to the PXO99, the strain with the Tal7b effector. So importantly for us to move forward, the MAGIC population contains lines with resistance and susceptibility to Tal7b. Now, it's not surprising that Tal7b showed that virulence function, that it had lines that it caused more disease, because we knew it was a virulence factor before we started, and we confirmed that in this study. So the bottom line on this one, in the GWAS analysis, we saw resistance to a RISE PXO99 with the empty vector. So the population shows resistance to the strain that we were using to screen with, not surprising. In fact, we know that this is the the XA5 locus, which Susan McCooch worked with a number of years ago. And that you can see those same loci were identified when we put the Tal7b into the strain. And also, we found that there was resistance that was unique to the strain carrying Tal7b. So we identified 2 loci that conferred resistance to Tal7b specifically, if it was in the strain that had 99, the strain PXO99. And also importantly for us, all these QTL or these regions were confirmed by interval mapping. So what's so interesting about Tal effectors? Well, there are different ways, and how is Tal7b potentially acting in this line? And can we predict something about how this resistance might be functioning by understanding something about Tal7b? So Tal7b, or Tal effectors in general, are known to interact with plants generally in 3 different ways. In one case, they bind to the promoter of what is called a death gene, a typical resistance gene type function, and they activate expression of that gene and you get cell death, something like a hypersensitive response. And the XA10, AVR XA10 interaction is an example of that. Another example of how Tal effectors interact with plant genes is that they bind to the promoter of the susceptibility genes, and when they bind, they activate expression of that susceptibility gene and you get increased disease. The hypothesis is, for example, that these sweet genes might be helping to deliver sugar out to feed the bacteria. The other hypothesis or other mechanism that's been demonstrated is where you have, instead of activation of a death gene, you have mutations or changes in the promoter or the nearby the promoter that don't allow the tau effector to bind, and you don't get activation of susceptibility, and therefore you have resistance by loss of susceptibility. So it's not a hypersensitive type of resistance, it's just the lack of disease. So we asked the question, how is the resistance to Tal7G functioning in these plants? Can we develop a hypothesis and predict what's happening by looking at this interaction? So if you look at a number of the magic lines as well as a control line, again, this is the empty vector, this is the overexpressed Tal7B, you can see that we have an enhanced susceptibility or enhanced virulence, enhanced disease, longer lesions in these lines that were not showing resistance. And this is most of the population, right? So this would suggest that the Tal7B is activating a susceptibility gene. It's one hypothesis that we can test. If we look at the lines that, and it's consistent with the whole population. If we look at the lines where we saw resistance, especially related to this QTL that we identified on chromosome 8 that conferred resistance to Tal7B, we see that there's no enhanced virulence. Tal7B is not increasing the virulence of 99 as it did on the other lines. And in this case, we would predict, or we would hypothesize, that we have either a change in the promoter region or nearby the promoter region that is where the Tal-B cannot enhance virulence because it's not activating the susceptibility gene. So the lines that have the QTL8 in them are consistent with a loss of susceptibility for the mechanism for resistance. Well, the beauty of the tau effector world is that you can predict what the target is in the plant because the tau effector code has been broken by Adam and other people, and we know how to predict what Tal7B should bind to based on the code. And in fact, using several bioinformatic tools that have been developed to ask what it binds to, Alejandra pulled out a set of putative targets that it might be binding to and activating in the plant. And as you can see, she hit on a lot of the genes that you would predict might be true. But she also hit on some other ones that are known to be involved in defense responses, but not necessarily had shown to be involved in these interactions. And these are genes that lie within the QTL that she identified. And one of the interesting ones, for example, that's not a sweet gene, is this SERC gene, because she sees a SNP in the EBE, the potential effector binding domain, that's actually close to the start, but not in the promoter, but very close to it in that particular gene. So I hope I've convinced you that MAGIC is a powerful tool for discovery of resistance and characterization of novel tau effectors. We screened these 330 lines, and interestingly, we were working with the S8 generation, which means that they had taken the lines and selfed them 8 times, 8 generations. We actually performed the analysis in an earlier generation, the S4 generation, and essentially landed on the same loci. So what that means is we maybe don't have to go out 8 generations to fix the lines and discover the things that we need. We identified novel broad spectrum resistance sources that target both XOC and XOO in those populations. And we also identified QTL that target a specific effector, 2 regions that target a specific effector, and we propose that the resistance mechanism, we have a hypothesis that we can now test, is that the resistance mechanism for one of those is through a loss of susceptibility. So the other important point is that all of these sources, you know, as a plant breeder, you don't necessarily care what the mechanism is. They're more interested in getting a useful and dynamic source of resistance that'll stand up in the field. And so now we can introduce these effectively into not into elite varieties, and then we can begin to ask the question, do they in fact increase the durability in the field populations, and are they broad spectrum at a field scale in the elite variety backgrounds? So I maybe talked a little faster than I intended, or a little slower, but I want to thank our many collaborators, especially our colleagues at, in Mali in Africa, who did the field screening led by Valerie Vidier from France, Alejandra and Anna here, and my lab group, along with Herman, who comes to my lab in the summer to get out of Texas, and many of you recognize him, and also to our many funding sources. Anna was supported by a Bechtel-Borlaug scholarship, and Alejandra by an NSF scholarship. So with that, I'm happy to take any questions. Yeah, Alan.
So can you look at transcriptomic changes, specifically like with the SERP?
Yes.
Versus the sweet gene.
Yes, and that's what she's doing right now. Absolutely, yeah. Because that's the next step, is to test which of those genes is involved. By the way, I neglected to say, I'm not surprised that we have multiple genes. I mean, we're talking about quantitative traits, and we've got 2 different ones. These TAL effectors could target multiple genes. In fact, they probably do. And so so I'm not at all surprised about that. In fact, I would bet that we will see more of that as we move through looking at each gene. And then she's doing that right now. She's got the lines growing and getting everything out to test the hypothesis. So it'll be really exciting. Yeah.
The field experiments in Mali looked interesting. How did you do the inoculation there? Was it the same infiltration and cut beef? But it didn't look like that. It looked like there was sort of natural inoculation.
No, in fact, it was inoculated. They had the pots, and they inoculated the pots from the outside.
Same way.
Yeah. Mm-hmm.
Same group of strains.
Same group of strains with a few exceptions. We couldn't put any Philippine strains in Mali. We could only put the Malian strains. So we only had 2 Asian isolates in our study anyway. So yeah.
You mentioned that the sequence or the locus was effective against both strains, but was against all individuals of both strains.
The one locus on chromosome 4 was effective at a statistic level against all. Not all, almost all. I think it was like 18 or so out of the 20, 18 or something like that. Probably the statistics, it probably showed some level of resistance. Remember, this is quantitative, and we're measuring lesions. The other loci were against a few strains of each. So that's why we're more interested in that one on chromosome 4. I don't know what to say about the ones that are effective against a few strains of Oryzae and a few strains of Rhizola, because the TAL effector complements in Oryzae and Rhizola are different. So I wouldn't expect that to be a reason. I would expect it to be something else. Yeah. Mm-hmm.
Janet, I was curious, your laboratory phenotyping, you were seeing resistance phenotype was lesion lengths, a third less, a half less, or whatever. In the field, is that resistance magnified much more in a field setting, or is it a partial kind of resistance?
It's a partial resistance. I don't know how it compares. I've forgotten. Yeah.
Epidemiologically.
Yeah. Oh, would it be effective in the field if it was just a partial?
More effective
Yeah.
than what it appears to be
Yeah.
on the lab assay.
Yeah. You know, I don't remember the numbers now, but I know that it was clearly resistant, more resistant, but I don't know how it compared proportionally. That's a good question. I'll have to go back and look at that data. Yeah.
Technical question.
Mhm.
So you mentioned about interval mapping.
Mhm.
Does that mean you create a new mapping population for that? Or?
You can do both with magic. Yeah, we use the same population. It's just two different mapping strategies with two different premises. So GWAS is a population. You're looking at sort of looking for a population, whereas interval mapping is, and again, this is where I'm the weakest. I'm not the plant geneticist, but in interval mapping, you're looking for association of a trait with a particular region of the genome rather than comparing across the population. Does that make sense? Yeah, yeah. But that's one of the nice things about this, because we can do both types of mapping because it's really a population, because you have 8 genotypes scrambled, so you can do both types. But you can't use the standard mapping tools that are developed for a biparental population. That's why we had to work with the folks that were developing the tools for the 8-parent populations. I should say this is an indica population, so those of you who know rice. There's also a japonica magic population, and there's what's called a global population, which is they took the 8 from the japonica and the 8 from the indica and crossed them, so 16 parents. Now, there are a lot of genetic problems when you put japonica and indica together, and so there's some problems with that population, but it's still useful for these kinds of things, identifying these kinds of traits, because it doesn't matter if you're missing parts of the chromosome didn't recombine. You don't really care if you identify something useful. So,
are there attempts to create artificial gene sequences that would increase the effectiveness of some of the genes?
well, interesting you should ask that. There are a couple, oh, I'm sorry. So you asked if there are attempts to create artificial gene sequences that would increase the resistance. So you notice, you may or may not have noticed, I took promoterome out of the title because I had another part that I was going to present that would have increased the length of the talk considerably. We know a lot about the activation of defense responses downstream of these events. And it's common, whether it be single gene resistance governed versus QTL governed. There's a lot of commonality of what's induced. Those are the defense response genes downstream. And a graduate student in my lab, Brad, identified conserved structural regions in the promoters of the genes that have cis elements that are conserved in defense response genes, several of them, and that if the genes are co-expressed, they often have these elements in them. And our hypothesis is that we can use those pieces of information either to edit genes so that they're better soldiers, they're better responders to the resistance genes, or we can breed genome-wide for more effective defense responses that are governed by these upstream genes. So yeah, I took it out because it would have taken a long time.
Sorry, Jen, I have another question. But,
Yeah.
So with the chromosome 4 QTL, I mean, if this broad spectrum, does that extend to like magniforza?
Well, you know, that's, if Anna hadn't had to graduate and leave the lab, that would have been the next steps too. She had a start on cloning the target genes there, because we've also got target genes there that we're interested in. And the other thing was to screen for magniporthy. So I've just come back from China on Saturday, and I've arranged with some folks there. I'm going to send it to them and have them screen, because we don't have the expertise for Magnaporthy in the lab anymore. And Rhizoctonia, I'm really interested, because we don't have single gene resistance for Rhizoctonia. So I'm really interested in whether, because it might, who knows? Yeah.
Yeah. Are there any blast resistance loci that are co-localized with that particular piece of the genome from other studies? I'm wondering, like, why there like new loci are popping out that the industry has yet to determine. Like, is it because those are new gene pools that are in the 8 parents?
It could be one of the, as I mentioned, it could be that we've created a new gene through recombination. That's one possibility, or that we've uncovered resistances that didn't show up in the parents because we've got them into new parent backgrounds. There may be, QTL. They can stretch across half an arm of the chromosome through the old mapping. So there very likely are regions that were identified for blast. This was novel for a rhizae and a rhizicle. There's nothing in that region. There are some single genes on chromosome 4, but not underneath that QTL. So it is novel in that region. And most of the resistances to single gene resistances to bacterial blight are not NBLRRs. They're not the traditional. To blast, there are a lot of NBLRRs. That region does have NBLRRs in it, so it's possible that we could have, yeah. But you just don't know. One of the things that we will also do, if I survive long enough to do it, will be to sequence those regions to see and compare them across the different lines to see what they actually have changed. But the SNP profiles clearly show that they're unique. So, yeah. Yeah, Andy. I knew you have to ask a question.
I have one. I'll save most of them, but and this is about mapping, so I know it can be a little challenging. But I'm wondering what the genetic resources are for the parents you have available, and how large structural variation at this region could impact your mapping locus, because you end up mapping back to Nicolare,
and IR64.
Okay.
Yeah. We have the SNP genotypes for the parents as well, but that's crude. We know we can predict which parent, for example, some of those QTL, we can predict which parent they came from. It's not trivial when you have 8 parents to do that, but we can predict because of if I can just quickly back up to this. If you go to Anna's paper, the full version of this is in there, and we can predict based on the haplotypic block which parent they came from, what that region came from. It doesn't mean it's identical to that parent, because it could have collected some small recombination that we don't measure with map. But we know by the the block approximately which region is contributing from which parent. Does that answer your question? If you look at Anna's paper, and I think it's in the supplemental information, that's made much more clear. Yeah.
Okay, well, as a token of our gratitude
Oh,
for your excellent and obviously thought-provoking seminar.
thank you. Wonderful. So, thank you.
This has been a production of Cornell University, on the web at cornell.edu.